everyone. Welcome back. I'm happy to introduce you to our next speaker, Philip Gribbon from our EU-OPENSCREEN partner site, Fraunhofer Institute of Translational Medicine and Pharmacology in Hamburg. Philip has a PhD in biophysics from Imperial College and worked at University of Manchester as a postdoc before joining Pfizer to work on fluorescence correlation-based high-throughput screening technologies. Since 2008, he has been involved in Fraunhofer Screening Port Organization, where in addition to experimental studies, he's involved in multiple European projects dealing with open research data and encouraging the full utilization of results by application of FAIR methods. In his talk today, Phil will explain to us why and how making your data fair is a great way to increase the impact of your research. Phil, as you can imagine, we are eager to listen to your talk as researcher awareness of the FAIR data principles steadily increasing. So I will give the floor now to you.
Thanks a lot. Kathy, can you hear me okay?
Yes, we can hear you.
Good, very good. Yeah, so I will just share my screen. Let me... Can you see my slides?
Great, we can see your screen.
Okay, perfect.
Great.
Good. Yeah, so thanks, Kathy. Thanks to the organizers for inviting me back this year. So I think maybe there'll be a few people who might have seen last year's presentation. And actually, this one is slightly different. The first part is the same, but the second part I've now dropped a lot of what I was talking about before in terms of some COVID-related projects. You've heard already about some of those from Heim today, and I'm putting in some information about the use of knowledge graphs and knowledge graph type approaches in drug discovery, and some examples we're doing there. So if we learn nothing from this the next hour, then this is probably the one thing that I'd like people to take into mind. You know, the situation we are in at the minute is with a limited amount of resources and that many of our organizations face. If we're constantly seeking to repeat the expensive experimental studies which have been previously undertaken, and we can't trust the veracity of data sets which are out there, then it makes it very, very difficult. So I think there's a responsibility on us all to be able to make sure that the work that we're doing at this point in time is available to sort of future users, and that we promote reuse of data sets. So drug discovery in itself is becoming increasingly, as it probably always was, a fairly data-driven exercise. But if you look at the numbers of papers which are coming out, which refers specifically to computational type drug discovery, which covers aspects such as structure-based screening, chemoinformatics, and pathway analysis, it's exploding at the moment. So tens of thousands of publications every year referring to it. Increasingly now, we're seeing the impact of artificial intelligence and big data and machine learning-based methods in drug discovery. And we've seen that with AlphaFold in the protein structure prediction, but also in target ID and validation, and helping to analyze complex data sets and biomarker profiles and genomic information and transcriptomic information in order to stratify patients for clinical trials, and also building models around the properties of compounds, especially when they're going to be going into animals and also the humans in terms of their ADME tox and PK/PD properties. So the AI methodology is being applied routinely now, and again, we're going to be a lot more papers in the last few years, and this will continue to be really an important area for improving the process of drug discovery in the future. Although it's not infallible, and there's a lot of hype around it, and we need to try and avoid that hype-driven aspects as well. Finally, just a quick note: there's new technologies on the horizon. The compute capacity provided by quantum computing is enormous, and I can see in the future there's going to be opportunities for applying those technologies, and we do have some projects here within Fraunhofer, although I won't talk about them today, where we're applying quantum computing methods to drug discovery. The other important trend currently is around the open science components. Open science is not just a simple thing. It's not just about open access to data, but also it's around the policies, the tools, the repositories, services, etc. So in the world of open science, which we're increasingly moving towards, especially encouraged by organizations like European Union and funders, then we need to think about a world in which we have to make our data and our results and the underlying data sets which led to the results that we publish in papers and we put in databases, making those available. And then you're going to get into things like reproducibility and replicability, for instance, and having all of the ontologies that you're using and all of those other guidelines and standards that go into your analysis being included as well. So open science is extremely important in this setting. And then again on the sort of open data aspect of things, I think there was some about 10 years ago, I think approximately, there was a number of papers published showing that a significant proportion of reported studies in high impact journals such as Nature and Science, etc., were not able to be replicated within and second labs or within industry labs, for instance. So if a new target had been identified, in many cases it was very difficult to then confirm the role of that target in the disease in another lab. And something around only 20% of data is sort of, in some cases, on the preclinical side. And that study showed that only about 20% could be easily replicated. As you also mentioned, around human-machine collaboration is extremely important in the application of AI methods, is going to be increasingly important. Experiments are expensive compared to compute power, but all AI does is essentially generate a set of hypotheses which can then be subsequently tested in an experimental setting. There's also data mandates set up by funders as well. I think that's also important. Funders don't want to be paying for the same study to be done twice or three times separately. They want the study to be done well, and those data to be made available through public funds. And then the publishing system is changing and a lot more open access. And then around, we need systems in place for recognizing those who are actually depositing data and making their underlying data available to the rest of the community. So recognition and credit for that. So if we look at a typical sort of leaky data pipeline within a PhD project or a postdoc project or any large project, a lot of data is generated during your process to optimize your assays, to bring together and understand how all the parameters interact until you get a stable protocol. And eventually, you'll get sort of useful data, but which may not be something which would go into a paper. Then you sort of process the data, and finally, only a small proportion of the paper actually ends up the data. So what we're talking about when it comes to reuse of data and fair data, in order to facilitate that reuse, is being able to see whether we can gain access from what was previously data that was discarded or wasn't made more widely available. And it's certainly the case that discoveries themselves are also enabled through the use of shared data. And that might be documents, SOPs, workflows, algorithms, etc., the data which is in databases. And that data has to be discoverable by humans and also machines as well. You've got to be able to access it and retrieve it in a standardized format so that you can reuse it, integrate with your in-house data. And then to a certain extent, the metadata which comes with that data has to be self-described so that actually you can understand how experiments were performed and then use that information in your subsequent analysis. Unfortunately, typically organizations may spend about 20% of their time gathering the data, but oftentimes about 80% is involved in data preparation and analysis as well. So a significant amount of our proportion of our time is spent analyzing data sets. So in order to address some of these issues, the FAIR concept came out, and this was originally a sort of gathering of those who are interested in FAIR, and I'll go into what FAIR means in a moment. And back in 2014, that developed into a set of guiding principles in a white paper, and then it became a publication in 2016, which is the FAIR guiding principles themselves. And this is a sort of a very highly cited paper now, and you'll see Many of these FAIR principles are starting to be are being adopted within academic organizations, but also within companies as well. There's also a number of SMEs who are just offering services around verification of data sets. So what does FAIR refer to? It's about making data findable, accessible, interoperable, and reusable. And if we sort of try and summarize that briefly, in order to make it findable, then you need identifiers, which have to be, ideally unique and resolvable, and you need those to retrieve and connect your data. You have to have descriptions of the metadata, so how experiments were done, how analysis were performed, in a community-defined way, and that enhances the overall discoverability. The terminologies that you use to describe the data should be consistent, so the same term should mean the same thing within different data sets. You need to understand the source of the data, who's responsible for generating the data, which organization, under which terms, and that contextualizes the data and facilitates the reproducibility. And then you have to understand what your underlying terms of access should be. So can everybody look at the data, or is it restricted to a certain number? What are the licensing conditions? So the FAIR means is the data needs to be as open as possible, but as close as necessary. And those terms of use need to be described in the form of licenses. And then through licenses, that allows valorization and also incentivizes innovation and reuse. So why do we need to be fair? I think it's really because the process of discovering a drug and further developing it and bringing it to the clinic is so incredibly difficult that you need input from many different organizations in a multidisciplinary setting, so from many different labs. So the information flow between labs needs to be enabled, and people need to be able to understand what's happening, what experiments were performed in another lab, and what results were, and how those can then be analyzed. So there's a flow of data across the individuals within the lab, within the projects, in organizations, and also going out to the wider community in terms of publications. And there's a flow across all the sort of tech infrastructure which exists to support that, various platforms, the databases and repositories, the registries of information that are being used there. And the idea is to reduce knowledge loss. And how do we create value from FAIR data and metadata? And you can sort of think of it in terms of the overall process of planning your experiment. So you plan your experiment at the beginning. You might have standard operating procedures that you refer to. They result in an experimental protocol. You have a number of processing steps and actually run the experiments, and you generate data. And then you within your experiment, you have a set of raw data. You may be applying quality control criteria to those data. So did your pharmacological control work? Did your standards work, etc.? You may then want to annotate those data, so information about which compounds you're screening, etc. Then you get to a point where you have your curated data. And then from that, you can apply analysis methods to those. You can derive information from those data, and that allows you to interpret the data. But when it comes to the overall value that you can gain from those, if you're able to have those data deposited in an internal archive, then that's a very good thing. That could be indexed. But the maximum value that can be achieved from working through all of these experimental processes and all these analysis processes, if you're able to sort of deposit those data into a shared repository, such as Kemble, and then those data can be used and reused by others, and then those data can also be indexed and identified and made findable. So the idea is to try and make FAIR a reality in a research ecosystem, or within a project, or within an organization or a company or an academic institute. So you need to have your concepts for how you're going to implement those FAIR data working. You need to build a culture of FAIR. So you're getting individuals, because it's individuals doing experiments. They have to answer the question, what's in it for me? Why should I spend all this time and effort making my data FAIR when it's going to be somebody else who will benefit from that? And from that, you need to create an ecosystem, so whereby people can verify and then be able to share and reuse their data. People need skills to do that, and understanding of what the processes are involved. And you have to introduce incentives for giving people a reason for actually putting in the time and effort to make the data fair, and then also metrics for understanding the fairness of the data. And that's this big investment in FAIR, and there's been a lot of work looking at and how you implement FAIR within organizations. But there's still a lot of questions. So what does FAIR really mean? Is this something, a concept which is just for the data managers and your database administrators? How do we do it in our lab? What can we use? Does everything have to be fair? Does every single experiment that I performed in this entire workflow, do I have to make sure that those data are fair? What do I do with all the experiments from my predecessor? And what about the legacy data? Who's going to pay for it all? And if I make the effort, who's going to benefit? Will I benefit? And how will I benefit if yes? So there's still a lot of questions, and it sounds hard. But if we want to sort of implement the fair data working, then we really have to spend time and effort investing in ensuring that we can identify different data sets, and this is using persistent identifiers, and also that we're able to cope with the needs of metadata. And that's providing contextual information, that's the application of standards and ontologies, and making sure that they're documented, and that's ensuring that eventually that the digital object that is produced through all of this work, the data, the code, and the other research object is available for reuse. I think one of the important things to remember is that better metadata makes better data. And you can see here, just as an example for the term that you use for the age of an individual who may be in a clinical trial, you can see there's so many ways of representing age, just in terms of the spelling, but then also, after birth, in years, year, etc. And that makes things incredibly complicated, especially when you want to use machine-based methods to reanalyze those data, and the context might be lost. So you can see just in this one example what the complexity is there. So in order to try and ensure that this metadata supports the FAIR process and the reuse of the data, we have to generate computer systems which allow for understanding with the relevant amount of accuracy in order to be able to exchange and integrate data set for an intelligent purpose, for a reanalysis purpose. And in order to do that, we have to consider the formats of the data, the terminologies which are used within the data, the guidelines, and also especially the identifiers. And we need to be able to create a conceptual model or a data schema which allows for exchange of data and for data to flow from one system to another. And that is helped and guided by controlled vocabularies and ontologies. We're using the same terms to refer to the same entity. We want to make sure that we've got enough information to describe what's happened. So we want minimum information, so that we've got a set of core essential information that helps us reuse the data. And then we have to be able to identify the data and its different elements through the use of persistent and context independent identifiers and schema. So there's a number of community standards which have been applied to data sets within our domain, either on the clinical side, such as the CDISC standards, or standards around proteomics or metabolomics or chemical data sets. And there's many, many standards for those, and it can be very, very complex and difficult to work with those. There's a lot of organizations which are involved on a European-wide level, trying to implement fair data working. I think there's the European Open Science Cloud, and some of the people on this call and others may be involved in European Open Science Cloud projects. And then there's also regional initiatives, different open science clouds within different areas. And then there's also a sort of a fair community involved in defining standards and helping provide training and guidance to individual scientists and scientists within organizations to ensure the implementation of FAIR as well. So there's a huge sort of ecosystem out there of organizations which are concerned with fair data working, and I think many of those are there to help. And hopefully, people in this training will be exposed to some of the outputs from these types of projects, and it will be able to help in actually implementing your experimental studies or your computational studies within your organization. And FAIR is moving towards being a new global norm. There's organizations like the Research Data Alliance and Go Fair who are sort of championing the development of standards. There's all sorts of policies, statements around, and FAIR's become the new de facto global norm, as it were, when it comes to data management. And if you're in a process, if you're involved in a European project now, you have to devise a data management plan which is based upon FAIR principles. So there's many organizations which are contributing towards the development of these principles. And then also to go along with the sort of policy side of things, there's also tools now which allow for individuals to be able to assess the fair level of their data, to get metrics for how fair their data are, and then provide guidance on how to improve that. And that's something which is sort of turning fair from an abstract concept to one which can be actually implemented within your lab, within your individual data sets. So there's a wide number of tools there, and I'm going to go through one of them in a moment that we were part of helping develop within the FAIR Plus project. So just to sort of summarize what it is about FAIR, so it's not a standard in itself. It's not one size fits all, and it doesn't just apply to a single domain. It's not about coming up with new protocols or something which is specific to any one technology, imaging or proteomics technology, etc. It's got nothing to do with quality. So you can have junk data, which is extremely fair. So you shouldn't confuse fairness with high quality. It's synonymous with open. It enables open science, but in itself, it's not intrinsically open as well. You can have fair data, which has a very limited number of individuals who are able to access it and reuse it. It's an architecture, it's a structure in which which enables our working and better data management and better reuse of data. And it's not written in tablets of stone. So it's a developing process. So it's really an aspiration. It's about a journey. It's ambiguous, and it's meant to be ambiguous. So it is complex. It's a standard set of principles which is being applied across all sorts of different scientific domains, and it's respectful of those domains. It should be implementable with today's protocols and standards, and a small part of it is around indicators. But really, it's about a framework for changes in the organizational culture and how organizations manage their data, and to allow them to be able to do that in a way where they can gain more value from that data. So if their data can be standardized according to community accepted standards for, let's say, a chemical structure, for instance, or a bioactivity, then they can, the value that it gives them is that they can then integrate external data with their own data much more easily, and then have a much broader view, potential activities that they're observing, and potentially make better decisions on the basis of that. And it's a work in progress. It's something which is sort of continually moving on. So it's a principle. It's not a precise practice. And there's a lot of people, who are looking at this. You need to, there is some concern about the amount of time and effort that's been involved in making data fair, and there's some guidelines coming out now about which are the data sets which should be considered to be ones where it's worth that investment to make those data fair. So we shouldn't make data fair just because it's going to be fair. There has to be a reason behind that. There has to be a driving business case to do that. And again, just to reemphasize, fair is not the same as open. They're slightly different, especially when you introduce aspects such as the GDPR, the European Health Data Space, and where you have regulatory elements which mean that a clinical data set may be fair, but by no means would it be open for everyone to be able to use it and analyze it. There's legislative and regulatory reasons why that doesn't happen. And there's also individual concerns about data coming out, people being scooped in publications, and it has a high impact on the individual's careers. But that's something that fair data shouldn't be posing issues there. It should actually help improve people's careers as it were. So one of the resources that has been created, hopefully that might be of use to folk on this call, is the FAIR Cookbook, and that's as the name suggests, it's a set of recipes to help analyze and understand the FAIR levels of data sets, and then provide specific examples on how those can be improved. It's an online resource, it's live, it's constantly being updated, and it's really for anybody who wants sort of practical assistance in verifying their data. And it was developed by a team of researchers and data managers, especially from the contributions from the Elixir communities, University of Oxford in particular, and also many of the other members of the FAIR Plus consortia. So you can use it as a researcher, as a software developer, data manager, or as a policymaker or funder, as it were. So the recipes are grouped around the different aspects of FAIR, recipes which involve findability, improving accessibility, interoperability, and reusability. And you can see them across there. And each is a kind of a step-by-step guideline around the particular topic. So you get the ingredients, the process, so what goes into it. So again, it's a recipe. So what are the different elements I need to consider here? For instance, around this example of ontologies, for instance, where do I find information about ontologies? And you can find that within the cookbook. And then also these are supported by practical examples and also examples of code, etc. And then further reading and how far you can go further with the FAIR Cookbook. So just Google the FAIR Cookbook and then you'll find it's there. And there's other recipe examples around, for instance, how one goes about generating unique persistent identifiers to annotate your data with and make your data more findable. So it was a lot of contributors to the FAIR Cookbook. You see some of them here. And also there's an ongoing editorial board, and members of our team are part of that from the Fraunhofer, and they're responsible for the ongoing maintenance and upkeep of the different sections. There's a couple of papers that have just come out which describe the FAIR Cookbook. I think they came out 2 or 3 months ago, and I'd certainly refer people to those. And then there's actually how you can then implement the cookbook is there. And then there's also webinars and other online information. So that's the sort of FAIR part covered, and these are some resources to help you. So you spent all this time making your data FAIR, and then you have to look at, well, what's the actual advantage in it for me? So I've got this FAIR data set. What are the benefits of having that FAIR data set? So that's the sort of second part of the talk. And I'll be talking about knowledge graphs as a tool which you can implement on FAIR data sets, semantically interoperable data sets, to be precise, which you can then use in the context of drug discovery and drug development programs. And I think, just as a bit of historical background, the original developer of knowledge graphs was by the mathematician Euler. And he founded graph theory back in the early 18th century. And the original question that he was posing himself was how he would be able to get across, walk entirely through the town of Königsberg, but in such a way that we'd only cross each bridge each time. So he developed a mathematical formalism to try and solve that problem. In the end, it actually turned out to be impossible, that you could visit all parts of the town only crossing each bridge once, but a new branch of mathematics emerged from that. But the way to consider it is that within graph theory, the neighborhoods of the different areas are considered as nodes. So that's where you want to get to. That's where the information is. And the bridges are considered edges. So that's how you get to that area. And this is something where we're working on a number of those types of projects. And one of the first sorts of things we want to do within this sort of knowledge graph area is to standardize your representation of your experimental data. And this is what we're doing in a number of projects where we're running electronic lab notebooks. So that's something that we sort of run internally. And within those electronic lab notebooks, we've standardized the representation of data sets. And those are access controlled. We share protocols and descriptions of how to analyze the data, which parameters are necessary to be recorded, etc. And those are shared between the different individuals involved within a project. Again, getting data ready so that they can be used in knowledge graphs. along with in some other projects in the it's focused on development of new antibiotics, so this so-called AMR accelerator. We've worked with innovative SMEs to develop software tools that allow for the recording of information related to in vivo studies on the efficacy of antibiotic compounds. And within these studies, we're able to record animal level results. So for each individual animal level, we can look at things like their exposure to compound, what the treatment regime is, what the route of administration, which bacterial strain they were administered, etc. We can group those into different treatment groups. And then we can analyze the results coming from there. So we can do, in this case, we're looking for colony-forming units, and we've got the log of the colony-forming units there. And we can look for all sorts of different types of experimental results and different types of study. And all of the data within this particular application, the way that we've developed it, is that those data are completely fair. They use standardized ontologies. And where we weren't able to access ontologies to describe these in vivo studies, we developed an application ontology of our own. What will happen with this data is that once a series of ring studies are being completed within that project, those data will be made available to the wider community. So all of this information at the detailed individual animal level, all of the data and all the metadata will be made available, along with the strains and the protocols running the in vivo studies. And supporting the process of using consistent descriptions of the metadata, so a consistent ontology. And we developed, or we extended upon the current bioassay ontology. We created a bioassay protocol ontology specifically to work with data sets from in vivo studies on to identify antibacterial compounds. So that's something that we've done and recently published. So coming back to the knowledge graph, so before is how do we get the data within projects in a format which can then be reused by knowledge graphs? As I said, there were first of all, as a kind of conceptual framework, it's from the 18th century mathematics that was done there. But since the 1970s, it's been used in database representations because it has got a number of advantages. It's able to have a structured representation of data, and it stores data beyond just sort of numbers and strings, and that enables the use of many concurrent users. And it can also handle multimodal data types. So you saw in some of the examples before, we were having information about bacterial strains, we're having information about animal models, we're having information about individual animal models and their housing conditions and their feed conditions and all of that different data. So there's lots of different data in there. And you could also have imaging information related to those data, or you could have sequencing information related to the strains that were being used. So it's got a number of advantages. They're widely used in many organizations. So when you go to Amazon, for instance, if you're interested, an individual, you've expressed an interest in cars and books and films and games, etc., and these sorts of things, they can then use all of these data in a knowledge graph approach and with machine learning methods to be able to identify what you're most likely to want to buy next. You can also have a similar type of thing within Netflix. If you have an individual A who watches a certain set of movies, if the profile of those individuals is similar, then for individual B, you can make recommendations which are the same. And you can see that on sort of a collaborative and content-based filtering as applied by organizations such as Netflix to make movie recommendations. We are involved here at the Fraunhofer in a number of projects with knowledge graph applications. So we're involved in the Buy COVID project, which seeks to provide a data infrastructure to support pandemic-related research and development, and create a set of data resources that can be used to bring together historical data related to the last pandemic, but also provide a basis for data sharing in future pandemics. So we've created knowledge graphs for monkeypox and Marburg, And we've those who are interested, you can have a look at some of the tools there. And we've been able to sort of pull out from public information all sorts of interesting information about the compounds which might be active within in monkeypox, the clinical compounds, their targets, etc., the pathways which are involved, both on the host side and also on the viral side as well. So that's a very interesting resource. As I mentioned, we built knowledge graphs around antibacterial data for this AMR accelerator group, and we created a pipeline for ingestion and standardizing project level data using the electronic lab notebooks. That's restricted to consortium members, but again, there's a subset of that. Those knowledge graphs are available on GitHub. And then we have another project, proxy drugs, that I'm going to show in a minute. In this case, we're talking about targeted protein degradation, where we've got heterobiofunctional drugs which hijack the ubiquitin proteasome system in order to eliminate proteins of interest from within cells. This is a new class of drug. It's a new modality. The compounds are just reaching the clinic. These are sort of bifunctional molecules, and we've created knowledge graph resources around these targeted proximity degradation. So we're looking at anti-infectives, looking at antibacterials, and also targeted degradation, and we've created these knowledge graph resources for all of those. And what does that actually look like? So this is the AMR knowledge graph. It's running in an application called Neo4j. And here we can focus in on a particular pathogen, Streptococcus pneumonia. And then this node here was a particular compound, and that was the properties of the compound linked through the Campbell. This is the assay that compound was screened in. There's another compound. These are all public data sources. We can then go into the particular publication where that compound was identified. All of these data are interoperable, so what we've done is we can combine public data with our in-house data as well, and then we can put those data into analysis workflows. And this is all easily navigable and linked into internal and external resources as well. And we've collected pretty much all of the information about all of the known antibacterial compounds, and then a number of sort of very important pathogens, and we've synthesized all those data. So all of those, essentially all public information on antibacterial compounds is present within this knowledge graph. We've also used knowledge graphs in the setting of COVID research. And in this case, what we want to do is trying to understand for rational repurposing, where we want to repurpose combinations of drugs, in particular drugs which may be antiviral drugs, so they act against the virus, but then also drugs which might facilitate the host and being resistant to the virus as well. And that's something which is quite important. If you can come up with an antiviral drug, then you can knock out the virus, but also if you can help the host as well in its infection process and reducing that infection process, then that's extremely important. So what we were able to do was to identify, using a pathway analysis, certain drug combinations of antiviral drugs and host-directed drugs, then look for synergism and antagonism and do some predictions around that. And then we came up with synergistic combinations of drugs that we were then able to test in virus-induced CPE assays. So could we see if we could hit the virus and the host cell simultaneously? And we were able to look at different combinations between, for instance, remdesivir, which is antiviral drugs, and nelfinavir and raloxifene, which, for instance, which is a host-directed drug. It's an estrogen receptor antagonist. And we can do dose responses and then look for synergies between them, and then come up with particular combinations, all based upon in silico analysis using knowledge graph approaches. I think one of the other important areas has been this targeted protein degradation. This is something that we're looking at as part of this proxy drugs project. One of the key things about targeted protein degradation is that it requires the presence of an E3 ligase, which performs in the E3 ligase in the complex, it performs a ubiquitinylation process. And there's only a few E3 ligases which have been identified up till now, typically cereblon and VHL. And there's an opportunity to be able to analyze large data sets and then come up with proposals for example, tissue-specific or disease-specific E3 ligases and compounds which are ligands for those E3 ligases, which can then be linked through a linker to compounds which are warheads for your protein of interest, and then you can eliminate your protein of interest in a tissue-specific or a disease setting-specific manner. So that's something which is extremely important for the targeted protein degradation world, understanding how the different linkers work in that, and then that can potentially help you eliminate targets from cells which are undruggable. For instance, transcription factors, etc., multimeric protein complexes. So what we wanted to do is to try and bring together all of the data so that is available within the public on PROTACs, molecular glues, and targeted protein degradation. And that's the activities within this Proxidate project. And we have, there's also a number of different mechanisms that we're interested in, not just PROTACs, but also molecular glues and lysosome-based targeting as well. Compounds that are involved in protein stabilization, cell therapy switches, compounds which could be involved in protein modification, etc. So we're part of a very large project, the Clusters Future project, where we've created a data infrastructure, and there's like a number of internal projects within that. So there's 9 internal projects which look at different elements of targeted protein degradation. You can see the different companies who are involved in there, and also Max Planck, Goethe University in Frankfurt, Technical University of Darmstadt, also, and the Fraunhofer. And what we did is we created a data infrastructure where we were able to manage all of the data within that project, again, using knowledge graph approaches. And so this was a collaboration between academia, Fraunhofer, and industry. And we were one of 11 projects involved in the data management of individual projects. And what that meant is that we were able to aggregate internal and external data resources related to targeted protein degradation in order to facilitate the identification of new E3 ligases, the design of new PROTACs, the design of molecular glues. And we implemented a fair data management process within that project. And that was something that really helped the project along. And within that, we took what was available from externally in terms of information about PROTACs and molecular glues. And there's a number of relevant databases, PROTACDB and PROTACpedia, and also from the PubChem databases. And then we brought in information on particular projects, information about our libraries, the assays, the data, the metadata, the compound structures, linking those through. Combining all of those together, information from particular projects like iGlue, which is a project looking for molecular glues. And then on top of that, we annotated with information around the targets that we're interested in from Drug Central, on the information about the E3 ligase system, on the UVNet information, the chemical information that we got from the compounds about the linker and the warheads, etc. And we bring all of those together, again, with data from open targets and Chembl. So we've got information about the mechanism of action of the PROTACs, the underlying disease, the associated side effects. And then from UniProt, we draw in information about the biological process involved, molecular functions, reactome, etc. And from that, we've created a knowledge graph with 9,000 nodes in there and 22,000 edges, pretty much summarizes all publicly known information about molecular glues and PROTACs. And it's all annotated with information from open targets, UniProt, Chembl, etc. And it's hosted within Neo4j. And it's also cross-linked to another database that in collaboration with colleagues at the IMG in Prague that we sort of set up as well. So whilst this information here, you can see all of the different parameters that we're able to store on that. We don't have a sort of a chemical information part. And this is something that we actually developed and was developed in collaboration with IMG in Prague. And they're using elements that they learned from developing the European Chemical Biology Database. And we're able to have a fair data repository for structural and activity information for PROTACs and molecular glues. And you can see you get information about structures, and you can separate the molecular glues into the E3 binding component, the warhead component, and the linker component. So you can look at the different functionalities there. And then you can also have a lot of annotation of information about the bioassays and the bioactivities involved as well. And that's all been done in a fair format. This is work done by Chibo and Toma and Petr Bartonek in Prague, in sort of helping us develop this for the proxy drug project. And we've got this centralized collection of all public data, and then we also appending proprietary data from the project within the project, so we can look at all of those data together. And then there's sub projects, and we each can have access to that. So what I'll try and do now is, I didn't want to do a demo, but we can show you a little video about proxy drugs. Hopefully you can see this, and you can see the resources they're using. So this is, yeah, so if we look at the proxy drugs DB, so this is the centralized database for consolidating the experimental data. So we can look at compounds, so we can sort of browse through the database itself. You can paste in your structure. You can look for substructures. You can do similarity searches, etc. You can find out which compounds in there are similar to your existing compounds as well. You can look at all the information that you have on your PROTAC, as well as the different components, what their properties are, what all the information which is in Chembl, etc. And drug bank we've got in there. And we can upload assay data, and this comes to the FAIR part as well. So this is similar to the ECBD. So this is the androgen receptor degradation. Androgen receptor is one of the most important targets in the TPD world, and there's currently phase 2 clinical trials in there. But we've been running screens against that, and we can include all sorts of FAIR data meta annotation of the properties of the assays that we've been running. We can look at the IC50s and other data. And then we can have different attributes. So is it what stage is it? Primary data, secondary data? What type of assay is it? So all of that information is in there. It's all stored in a consistent way using accepted ontologies. And then that can be integrated with all of this public data that we've pulled in and put into a knowledge graph. And then this is the knowledge graph itself, which is the PROTAC KB. So this is the androgen receptor. And these are all the different properties, the mechanism of action, disease, biological processes. So you can look at all of these links out to disease that it's associated with, names, other links like PubMed, Open Targets, UniProt, etc. Other descriptors there. You can look at the E3 ligases involved in PROTACs against the androgen receptor. You can look at the different targets involved. Pretty much everything. So this is all the data that we have. These are the different warheads which bind to it, the different E3 ligases which are involved with those warheads, the different assays which are involved, all around the androgen. So we have like all of the information on androgen receptor TPD properties sort of contained within there. And we can look at the individual PROTAC itself, which binds to the androgen receptor. We can look at the proxy drugs DB thing, where it is in Chembl, and then we can actually look at the structure. So we can link out to the structural information from the knowledge graph. So all of this data is integrated and linked as well. So we also have a search interface, so we can look for specific subsets of data, specific relationships. We're using this Cypher query. So for instance, give me a certain warhead with a certain E3 binder, give me all the PROTACs for a particular cancer, for instance. So that's the sort of a final part of it. We're also using those data as well, and but that's something else for another day. So just to finalize the last two slides. What we have here is everything I've been talking about in the last hour I hope it's not more than an hour, but in the last hour has been around preclinical data and discovery data. But all of the sort of essential principles also apply to clinical data as well, but with the added complexity associated. I'll just stop the music on my side. Can you? Yeah, but with the added complexity which is associated when you're working with sensitive data sets and work with clinical data and all of the regulatory and other problems that are involved in there. So I'm now part of there's a large innovative health initiative project called IDERA that's launched this year, and we're one of the leaders there. And within IDERA, we're trying to address the fairness of clinical data and whether we can use fair clinical data in order to support the development of machine learning-based algorithms to help improve the process of detection, diagnosis, and prognosis and ongoing treatment of lung cancer patients. This is a very large project, so 200 people involved. We're going to create an open platform. It's going to be about allowing for enabling connectivity, access, sharing, use, and reuse of health data. And from that process of showing the value of those different data, of working with those different data sets, we want to try and change the policy making, decision making around how you use data within the clinical trial setting. So if you run a clinical trial where one of your endpoints is a machine learning or an AI-based generated endpoint, how do you understand what's gone into making that reaching that final data point? And what the other considerations are in terms of the believability, the robustness of that, the ethical compliance of that, the coverage of that. So is your training set competent to be able to actually be used as a model for your test sets for individuals who are in different countries and different settings, etc. So there's a lot of different partners in there. It's led by ourselves and Johnson & Johnson, along with Sanofi and Takeda and patient organizations. And we look in regulatory acceptance. We're also very interested in standards and data management. We're creating an architecture. We're interested in real-world evidence, public health management, lung cancer, ethics, patient-reported outcomes, etc. And the way that it works in IDERA, again, exactly all of the same principles that I've talked about earlier in this preclinical setting are transferable over to the clinical setting. We want to be working with fair data, but it's much more complex data. So it's electronic health records, it's imaging of patients using CT scanning and MRI, et cetera, using these DICOM standards. It's wearable technologies like watches. It's data coming directly from clinical trials. We've got a number of use cases which are around care and around research. We're building a federated infrastructure in a privacy-preserved setting, so we're not pooling data, but we're keeping the data at its source, and we're sending the analysis to the data set itself, and we're building a set of connectors and a lot of services around that, and we're going to be using those for risk prediction, algorithm development, and remote patient monitoring, and we're also interested in connectivity, and eventually getting to a point where we can use these findings to justify the continued use of AI methods in clinical data. So that's everything. So it's quite a lot, but just to reinforce, there's a lot of people involved in this, all from different teams, especially from our team here, but also from support from EU-OPENSCREEN, and especially the colleagues at Prague at IMG. But these are some of the people I've been working with, and especially the FAIR Plus and Elixir teams who really developed all of the FAIR framework, and we were part of that. So that's everything for now, and I'm happy to answer any questions.
Thanks, Phil, for this interesting talk. I believe that this is a topic that we cannot cover in one session. So with your help, we are going to discuss it over and over again. Yeah, we have many questions, but only some time to answer them. So I hope you can also see, Phil, the Q&A box with the questions. Okay, great. Let's go to the first one. Is it possible to leverage the power of AI machine learning-based analysis to solve the complex problems inherent in drug discovery?
Yeah, I mean, that's a good question, and there's a whole set of papers I think appeared recently. There was the Journal of the Journal of MedChem, there was a paper saying, why isn't AI being used so much in chemistry? And I think it's essentially because the training data sets don't exist. So all of these things I've been saying about fair data, etc. If we want to start using these methods, then we need to have other people who are generating hypotheses and applying their algorithmic tools. They need access to high quality relevant data sets to act as the training sets. And I think it will be possible. A lot of hype there. There's a lot of companies who are sort of setting themselves up as we're the AI discovery company, but many of those are finding that they actually have to invest in experimental resources to generate data sets themselves. Companies, big pharma companies, and I've worked in one in the past, they have access to a lot of data, but the reality is that much of this data is unstructured. Most of the data doesn't have the right metadata, so you don't really understand what was done, how it was done, and that's essential in being able to develop an algorithm and apply an algorithm. So yes, it can be used, but at the moment, we're hindered by the lack of fairness, which is why I started off. The fair part today is the important part, and that's the one that's boring and involves a lot of time and effort, and nobody can really see what's in it for them. But actually, there's a huge amount in it for them if they can use these algorithms for the benefit of their own projects later on.
Great, thanks, Phil. Let's go to the second one. Human errors during data entry and potential bias in data collection can affect the reliability of research findings, impacting drug development, patient safety, and regulatory approvals. With the FAIR principle, how can you avoid potential selection bias for using stored?
So I think, as I mentioned in one of the slides, FAIR has got nothing to do with quality. Yeah, so they're two completely different things. So if you perceive errors in human errors or other errors, you can make the error in the selection of your which fluorophore that you're using in your assay, because it then interferes with your compounds in so many cases that it drowns out your signal. The compounds drown out your signal. So there's lots of types of errors. I think that within the IDERA project, we have a work package which is pretty much dedicated towards patient understanding data quality. So there's a whole set of frameworks around which are being currently developed to try and understand what the quality of the data is and what the competency of that data is. And especially in the case of drug discovery, and this is something that we're also seeing in the PROTACs world, is that you have it's a very sparsely populated matrix. So once you get beyond the initial, you've got a compound structure, you can then compute multiple parameters of that, the TPSA, the C-log-P, etc. You can, once you've got a structure, you can compute parameters. Once you've got a compound, you synthesize, you can put it in bioassays and you can get biological results. Yeah. Once you've run your assays, so that's a certain data set. Once you've run your assays and picked out your priority compounds, you can optimize them and put them into ADME-tox, and you're getting less and less, you're getting more and more data on less and less compounds. Yeah. So if you want to, for instance, build a model around to predict the pharmacodynamic properties of a compound, you typically have very, very little data in order to do that, because only a small subset of compounds have reached that stage. And then, and those data typically are not available within public resources or repositories like Chembl, for instance. So you get to this point where you don't have a problem with compound quality, but you don't have a problem with data quality, but around the sort of the scarcity of the data that you actually have, and then you have to then impute and fill in the gaps, as it were, in order to get your model to run successfully. So it's like quality, lack of data, lack of fairness are all factors which impact, what you can do, and that's relevant to both the clinical setting and the preclinical and discovery setting as well.
Great, thanks, Phil. Next one, technologies that were used in previous research may no longer be supported, or individuals who were responsible for creating the original data sets may have moved organization. What can be done to reduce the incidence of trapped historical data?
Yeah, so that's So that's a very good point. Is instrumentation moves on in terms of its throughput, in terms of its sensitivity, in terms of its sort of range of applicability. So your mass spectrometry system from 20 years ago is in no way comparable to the sensitivity that you can get from a modern day quadrupole mass spec, for instance. So should you, and that gets to the question you need to ask yourself when you're looking to verify data: what do I potentially gain from doing this? And there'll be many situations where technology's moved on, and it's much better to invest time and effort in making sure that your modern processor, your current processor, are up to date, and you have a sort of fair at birth type concept where the data is immediately fair. And at that point, you need to be talking to instrument creators and the people who are writing software tools for analyzing data sets to see whether we can actually improve the fairness of those data sets that are being generated. Now, typically, it's not worth going through lots of historical data in order to make it fair. That's what we've seen. I was in the FAIR Plus project. We were responsible for verification of 20 data sets from different IMI projects, and it was very, very unlikely that we were looking at really historical data sets. There's a minimum that they're machine readable, but typically technology's moved on from that. So it's a question of degree, and you have to make decisions about where you want to put resources. And typically it's better to put resources and make sure your future data is going to be fair.
Thanks, Phil. So last question, can we deploy smaller data centers in the future for storage?
Yeah, so I think storage is not the issue. I think what you need to look at now is this thing coming up called the European Health Data Space. And that's going to be a set of sort of legislations and rules that will be applied within Europe to govern how individuals are able to sort of give access to their data for secondary reuse or for primary use, or secondary uses for research, for instance. So I think instead of smaller data centers, we're going to have this concept of a trusted research environment, which has all of the necessary security systems in place. They're compliant with regulations. They have the infrastructure for storing the data, but also for working with it in a secure way. And I think that's going to be more the future. So it's going to be these trusted research environments, secure processing environments are going to be the infrastructure model of the future for working with, especially with clinical data sets. And organizations like Elixir and Fraunhofer and others are trying to create those, and also create the governance framework for people to be able to deposit their data there, whether it's clinical or other data types, or people to be able to access it and run their algorithms that they've established on those data sets.
Many thanks, Phil. I hope we will have the opportunity in the near future to discuss more about it. Many thanks again for your participation in our school. And yeah, have a nice day.
Thanks a lot. Thanks.
Bye. Bye. So yes, now we are about to have our lunch break. So please stay tuned, because our last session for today is from Mabel Loza and Maria Matos from the University of Santiago de Compostela in Spain, our Spanish partner site, who will give a general introduction to chemical biology. So we will be back at 2 o'clock after our lunch break for our last talk.